Good morning. Good morning and welcome to day 2 of the Australian Dementia Research Forum. I'm Peter Schofield. I have been involved with ADNeT since the original grant application and also one of the founding members of the ADNeT Limited company. Currently, I must have been very bad because I'm the interim chair of that company as well. And the ADNeT Limited company has been the vehicle through which we've channelled Department of Health grants and industry funding and channelled it out to various participants of the ADNeT network. So it really has been a pleasure to be able to help with achieving that on behalf of everyone in the dementia research community. So I hope you all enjoyed some outstanding sessions yesterday. Stephanie, the acronym was HOPE. I've forgotten H. Orals, posters. Hello, sorry. Because you had to, you see, I've half got it, but I'm trying to show that I was here and I did listen, even if I've only half remembered. Because you have to meet new people. And that's the exhortation for today. You have to go to an oral session that is not in your comfort zone and learn something new. You have to talk to a poster presenter and encourage them with their work, and the exhibitors. And you have to go and be lovely to our exhibitors who are also our sponsors because they have both technology, support for the sector, medicines, diagnostics, all things that you should be able to use. So one of the really lovely components of a session like this is to recognise through the ADNeT Lifetime Achievement Award an Australian dementia researcher who has made outstanding contributions. And it could be in science, in clinical care, in community. It is the generality of contributions. And so today I am delighted to be able to present that award to Professor James Vickers. Now James can stand there while I embarrass him for a moment or two. So James is a distinguished neuroscientist and, as most of you will know, the former director of the Wicking Dementia Research and Education Centre at the University of Tasmania. Over many years he's made an extraordinary contribution to dementia research, to education, to prevention, to community awareness, both in Tasmania and nationally. And if you came to Jane Alty's keynote yesterday, you would have seen parts of that legacy that James has established. James personally is internationally recognised for his research in neurodegeneration and brain ageing, particularly understanding mechanisms underlying Alzheimer's. His leadership, the Wicking, became nationally and internationally respected Centre for Dementia Education and Research. One of the crowning achievements that they have achieved is the development and dissemination of their MOOCs, the massive online open courses, across initially dementia, but now in so many other neurodegenerative conditions. And those MOOCs have reached hundreds of thousands of individuals. And what a fantastic way of raising skill and awareness in both professionals, but also the community. James has been a passionate advocate for dementia prevention, public engagement, research translation, and of course, in his role as the director of the Wicking Centre, mentoring and developing emerging researchers and helping to build Australia's dementia research capacity. So on behalf of ADNeT and the broader dementia community, we thank you, James, for your contributions, for your remarkable leadership, vision, dedication to improving the lives of people affected by dementia. So please join with me in congratulating James on receiving the ADNeT Lifetime Achievement Award. So, do the right thing. I'm going to take that back. And I'm going to open up the trophy, but you've got to promise not to let it fall out of the box.
There we go. Thank you very much.
Keep a hand on it. I'll take that. If I put that there and there, then I'm going to hand the stage over to James for a couple of words.
Thank you very much, Peter, and many, many thanks to ADNeT for this fantastic award. Peter will not remember this at all, but on the topic of meeting new people, it was actually my first Australian Neuroscience Society meeting back, I think, in the late 80s, where we were these sort of brash young PhD students, and we came down for breakfast wherever the hotel was, and at our table was Peter Schofield, and we were very full of ourselves, so we were describing our wonderful projects where we had really done absolutely nothing and published nothing by that stage, but we were pretty full of it. And then there was Peter at the table, and we said, oh, well, we thought you were another PhD student, so fresh-faced and young-looking. And we said, oh, you know, what did you do? And he said, well, I published a paper in Nature on cloning the GABA-A receptor subunits, which was, you know, a monumental achievement in neurosciences. So that was humbling, and put the bar up there for us as well. So thank you, Peter, for your very kind comments and for being there at the beginning and now at the end of this career. So again, thanks to ADNeT. It's been wonderful to be part of ADNeT. I'd just like to say a few thank yous if I can take up a little bit more time. I want to put these in, I guess, three groups. But first, and really foremost, really to my family. Luckily, my wife Charlotte was able to come along to the meeting today, which was fantastic. And our children Beatrice and Thomas have been steadfast supporters over the decades and very forgiving of the times when their father had to spend time writing grants and going to conferences, etc. So I'm very grateful for that. We also know too that when you get an achievement award really of any kind, it's not really about you doing all the work, it's really you plus all of your colleagues. So I'm really grateful for all of those people I've worked with over the years, the research assistants, technical assistants, admin staff, research high degree students, research fellows, other academics in Tasmania and beyond too that we've collaborated with and undertaken a number of projects in the educational and research space. So my deep thanks to everybody for being part of that journey. In particular, I'm really deeply honoured to have been part of the Wicking Dementia Centre. It was a particular honour to be the director. This was a centre that was co-founded also with my dear colleague, Professor Andrew Robinson, and it really has been for the last 17 years or so a wonderful place to work where everybody showed commitment. It didn't really matter what discipline was your background, we all came together to try and make a bit of a difference for people with dementia and those that care for them. So my thanks to all Wicking people in the audience today around that. And then lastly, a lot of our projects involve a substantial amount of engagement. At one end we have our own clinic where people come in, and on the day they probably get some pretty dreadful news, they're still there to volunteer and be part of research. So I think that's just been a fantastic thing. Just about everybody who comes to our clinic is involved in research in one shape or another. We've had a number of community programs that involve, in particular, the Tasmanian population. Many thousands of Tasmanians have contributed to that research and stuck with us over the years as we've drawn their blood and put them through their various research paces. And so again, we're very grateful that we've been able to work so closely with the Tasmanian community. And then beyond that, of course, in the educational space, we've worked with a large number of groups throughout Australia and overseas in developing those MOOCs, and we've also been really grateful for the uptake. It's just every time we run one of these MOOCs, they're really a great sort of social learning enterprise, and many of the people who do the MOOCs give us plenty of useful feedback, which we try and incorporate in further iterations. So we're grateful for everybody who's engaged with that. That's about it for me, but really I am retiring from my formal university jobs, but I hope to maintain some research interests through an honorary position with the University of Tasmania, so you haven't quite got rid of me yet. But in the meantime, I know it's difficult times around Australia, and despite having a National Dementia Action Plan, there really just is not enough funding around to help support all of the fantastic research projects that we have going on in Australia, and that's something that really needs to be addressed. So I encourage you to keep at it and to keep doing that wonderful work that's going to make a difference for people with dementia now and into the future. Thank you very much.
Well, congratulations, James. You started off saying that you were full of it, but I really salute you for not only your tremendous research achievements, but what you've done for education in terms of dementia throughout Australia. And I find you the most humble researcher I've ever known. So thank you. Let's please congratulate. Good morning. I'm Professor Ralph Martins, and I will be your chair for the 4th keynote of ADRF 2026. And it's my pleasure to introduce Professor Brendan Lucey, who among sleep researchers throughout Australia, he's very, very highly regarded. And Sharon Naismith, unfortunately, who couldn't be here today, is a close collaborator, as well as many of our other sleep researchers. Professor Lucey is a professor of neurology and director of the Sleep Medicine Center at Washington University School of Medicine in St. Louis. He is internationally recognized for his research into sleep, aging, and Alzheimer's disease, and his work has been supported by the National Institutes of Health, including a K76 Paul B. Beeson Emerging Leaders Career Development Award in Aging, as well as funding from the Bright Focus Foundation, the AASM Foundation, and Open Philanthropy. Today, he will present his keynote address titled Translating DORAs into Disease-Modifying Therapy for Alzheimer's Disease. Please join me in welcoming Professor Brendan Lucey.
All right, good morning. Thank you very much for the invitation. I'm honored to be here speaking with you today about some work we've been doing translating some basic science findings around dual orexin receptor antagonists to human use to try to see if we can modify AD pathology using these medications. I am currently at Washington University, but in a few weeks I am transitioning to the University of Minnesota, and that's why I have the other label up there. These are my disclosures. The main thing I want to talk about is that I will be talking about some drug trials using suvorexant and lemborexant, and drug is provided by Merck and Eisai respectively, and those trials are looking at some off-label use around AD prevention. I just wanted to, go over, I'm sorry, I thought I was on the wrong slide for a second. I'd like to just do the brief overview of what I'm going to be discussing today. I'll do a brief background about sleep and the orexin system. I'll then go over some of the publications around the preclinical findings using DORAs in AD and some human studies. Talk about some recently published data in a preprint doing some cross-species translation, and then go over some unpublished data about plasma AD biomarkers and proteomics, and then end with some future directions. I just wanted to do a brief primer for this audience about sleep and about the orexin system. I think we all know that sleep is a rapid reversible behavioral state where we're not as responsive to our environment. It occurs with a circadian distribution, meaning we obviously sleep at night ideally and are awake during the day. And as we maintain wakefulness, we build up a sleep need or a homeostatic sleep drive. And these two processes work together, where there's the process C, the circadian process, the internal clock that's promoting wakefulness during the day, and then at night is promoting sleep, and then the process S, which is that homeostatic sleep drive, where your need to sleep builds up with prolonged wakefulness. The orexin system does a number of different things in the body. I think about it most of the time around sleep-wake activity, but it actually receives inputs from numerous systems in the body and has outputs to numerous brain regions. Orexin neurons are located in the lateral hypothalamus, and they receive signals regarding metabolism, energy homeostasis, as well as circadian inputs from the suprachiasmatic nucleus. And then they project out to arousal centers in the brain to help promote wakefulness. So orexin peptides, orexin A and orexin B, are wake-promoting neuropeptides in the brain. And when orexinergic neurons are inhibited by the VLPO, that helps to promote sleep. And one way to think about the orexin system, at least in terms of sleep-wake activity, is it's kind of like it's sticking its finger on the scale of wakefulness. You know, when your brain is seeing that orexinergic input, it's helping to promote wakefulness and to stabilize those sleep stages. Narcolepsy type 1 individuals are deficient in orexin, and so they have very unstable sleep-wake activity, and they kind of teeter-totter back and forth. So a key preclinical study that came out in 2009 that certainly has influenced my career in this area was this science paper from David Holtzman's lab at Washington University, where they showed that soluble A beta in interstitial fluid oscillates with sleep-wake activity. And that's the figure that's furthest to the left of the screen. On the top is the concentration of interstitial fluid amyloid beta rising and falling with the minutes of sleep per hour. And they showed that you could manipulate this system by using sleep deprivation, which is the next slide over. They kept these mice awake, and the soluble levels of amyloid beta increased. And if they gave orexin, which is wake promoting, they also could increase those levels. Whereas they gave a DORA, or a dual orexin receptor antagonist called almorexant, they were able to lower those levels. And when they gave almorexant for a prolonged period of time, in this case 8 weeks, they were actually able to reduce the amyloid plaque burden in these transgenic mice. So the top figure A, that's the control where they had sleep-deprived animals versus normal sleep. And in the black bars, those are the sleep-restricted animals, showing that they have a lot more amyloid accumulation with prolonged wakefulness. And the bottom graph on the left is the almorexant versus vehicle control animals. And you can see in the black bars, the almorexant group has marked reduction in amyloid plaques in multiple brain regions. And then on the right side of the figure is another paper from David Holtzman's lab where they knocked out the orexin gene in these APP transgenic mice that develop amyloid plaques and markedly reduced amyloid plaque formation. So I've undertaken a number of translational studies in terms of my career journey. And the first one we started with was looking, could we just do sleep deprivation to change the levels of these proteins? And we looked at different forms of amyloid beta. We also looked at unphosphorylated tau. And so in this slide, it's a lumbar catheter study in cognitively unimpaired, biomarker negative participants. They were 18 to 60 years old, and they came in the morning and they had a lumbar catheter placed on their lower back so that we could collect cerebrospinal fluid every 2 hours for 36 hours. And so the catheter was placed at 7 in the morning. They were all kept awake until the vertical dashed line, and that's when we allowed them to sleep. And so they were all under the same conditions. The lines overlap at that point because we took the average of the first 12 hours, 7 a.m. to 7 p.m., and then normalized all the data points across the 36 hours. So at that vertical dashed line, the participants were either allowed to sleep normally or as normal as they can in our unit with a catheter in their back, and someone coming in every 2 hours to collect samples. And that's the blue line. The red line is the sleep-deprived participants. And then the green line was a drug group that where we gave them a GABA B agonist called sodium oxybate, which is a medication that increases slow wave sleep and is used to treat narcolepsy. And there was no difference between the control and the drug groups, but in the sleep-deprived participants, we had an increase of amyloid beta of about 30 to 50% compared to the controls. And we saw very similar findings with forms of unphosphorylated tau, so tau 181 and tau 217. And the shaded area is the sleep period. There's a well-described delay from when we see changes in the brain reflected in the lumbar catheter, and so that shaded area is when all the participants in the study were asleep. I was very interested in looking at plasma AD biomarkers. I think this is a really hot area in the field for obvious reasons. I was interested in it because I wanted to see if I could stop doing lumbar catheters and just collect their blood. The answer to that was no. If you look on the left side of the slide, there's, this is from that same sleep deprivation study. You can see CSF A beta 42 and CSF unphosphorylated tau 181 showing the findings that we just looked at from the sleep deprived and control participants. But when we look in their blood, we actually see the opposite effect. The control group is above the sleep deprived group. We interpreted this as the sleep deprivation was impairing clearance from the brain to the periphery. And that's why we see higher levels of amyloid beta and unphosphorylated tau in the cerebrospinal fluid in the sleep-deprived participants, but lower levels in the blood. We also looked at phosphorylated tau, and that's the right side of the screen. We looked at phosphorylated tau 181 and phosphorylated tau 217, and both of them were increased. But 181, interestingly, wasn't any different from the unphosphorylated form. So in the bottom is the ratio, and those are flat. There's no group differences. Whereas 217, it actually was increasing. Sleep deprivation was increasing phosphorylation at this site, suggesting that sleep deprivation is not only increasing the levels of these proteins, potentially by reducing their clearance or their production or release, decreasing their clearance, but also affecting phosphorylation at certain sites. About the time that study was kind of getting toward the end, the first DORA was approved. That was suvorexant in late 2014. And so I thought there would be a great opportunity to do a direct translational study of that mouse experiment that used almorexant. And since that time, there's been two other DORAs that have been approved, lemborexant in 2020 and daridorexant in 2022. And so for this translational study, we recruited 45 to 65-year-old participants, again, cognitively unimpaired. They were biomarker negative, so no amyloid accumulation. Our goal was to have 15 per group. We got to 14 before COVID kind of disrupted things. We had people who dropped out for a variety of reasons. And the basic study design was very similar to the previous catheter experiment, except that we had them come in at night. And so the catheter was placed about 8 o'clock at night. An hour later, they got their first dose of drug or placebo, and then they were monitored overnight. You can see the first sleep period, kept awake during the day. They got a second dose of drug, and we had another sleep period. And so it was another 36 hours of sampling via the lumbar catheter. And what we had found was that the suvorexant 20 milligram group, which was the highest FDA-approved dose, reduced A beta levels across all these different isoforms. We looked at A beta 40, A beta 42. We also looked at A beta 38, but it's not shown here. To follow these columns of graphs, if you look on the far left, that's the concentration of A beta normalized to hour 0. And then the middle is the percent change, so it's normalized to hour 0. And to account for that delay, the right side, those figures show normalization to hour 6, which is about 5 hours after they receive their dose. And you can see that the concentration of A beta is pretty flat from that point on. This was in many ways a very unexpected finding. The lower levels of amyloid beta are persisting into the afternoon, 2 to 4 p.m. I'd originally hypothesized that we would see the reduction, in the late part of their sleep period and then into the early morning after they were awake, given that delay. And I was surprised to see it stretch out for so long. And I regretted not doing 78 hours of sampling, because if you look at the far end of the graph, the suvorexant 20 milligram group in green is remaining relatively flat, while the red placebo group is starting to go up again. And I wonder if another 12 hours of sampling might have revealed even more differences between the groups. We also looked at forms of phosphorylated tau. The big finding was tau 181, whereas if you look at the top, we have the left column is just the ratios of the different phosphorylated tau forms. The middle one is normalized to the hour 0, and then we have the area under the curve across the entire time course. And the major finding was that the suvorexant 20 milligram group in green was reducing the p-tau 181 levels pretty quickly and maintaining them lower than the placebo group, so that the area under the curve across the entire 36-hour period was lower in the suvorexant 20 milligram group. The other forms of tau, we didn't see any, phosphorylated tau, we didn't see any differences between them. The 202 group, I wonder with longer sampling if those curves might have separated more. And this raised an interesting question of what's going on. We didn't see a large effect of sleep with this drug. I don't think this is very surprising. These are participants who are brought into a new environment. They have the catheter in their back. They have someone coming in to collect samples every 2 hours. But we did not have any statistical differences between the different groups for total sleep time or sleep efficiency, time in different sleep stages. And I did wonder if this was maybe a signaling effect. Orexin signals through a G protein coupled receptor. It's been associated with ERK signaling, for instance. And could there be changes in orexin signaling that are having a more persistent effect on amyloid beta, a phosphorylated tau, that go into that waking period? And David Holtzman's lab recently published a paper looking at lemborexant, another one of those DORAs, and its effect on tau-mediated neurodegeneration. And I think really importantly, they actually compared it to another sleep drug. They compared it to zolpidem or Ambien. And so you can see in the hippocampus and in the entorhinal cortex of mice, the vehicle control and zolpidem at Zeitgeber time 13 and lemborexant at Zeitgeber time 3 and 13. And what Zeitgeber time is the time from when there's the start of sort of the circadian day by the behavioral intervention, in this case, lights on. So Zeitgeber time 13 would actually be at the start of the dark period for these mice, and mice are going to be more active. And whereas Zeitgeber time 3 is going to be when the lights have just come on and the mice are more likely to be sleeping. And lemborexant has reduced tau-mediated neurodegeneration compared to the vehicle control, as well as zolpidem, although there are differences depending on the time of day when it's given, which I think makes a lot of sense. The Zeitgeber time 3, that's going to be when the lights are on, when they're sleeping. It's kind of promoting their sleep during their normal sleep period, whereas Zeitgeber time 13, it's like they just had their sleep period, and now you're giving them a drug to make them sleep even more. And they were able to show that this effect on tau-mediated neurodegeneration was due to changes in PKA-mediated phosphorylation. They also looked at ERK and P38 and showed that lemborexant reduced the activity of those phosphorylation kinases compared to zolpidem or the vehicle control. Another potential, this was just published, I think, a week ago out of Eric Music's lab at Washington University, as another potential mechanism that a DORA might work. And so this is using lemborexant and looking at the effect on microglial clearance. So on the left side, basically what they showed is that there wasn't a reduction in the number of microglia, but lemborexant increased the number of activated microglia. and this was associated with increased clearance of amyloid plaques. They did some transcriptomics and showed that there was increased lysosomal and proteasomal activity that may be driving this effect. So these studies in some ways raise even more questions about how DORAs are working to affect AD pathology. And I've been very interested in this translational space and how to move back and forth between preclinical animal models and human studies. And so I've been collaborating with Douglas Brubaker at Case Western Reserve University, who has a system called translatable components regression, or TransComp-R, where they use publicly available data from mouse transcriptomics. In this case, they looked at 4 different animal models and are able to find the human homologs and sort of test what the effect would be of different interventions on the mouse homologs of human genes. I wasn't involved in the modeling. It's mathematically quite a bit above my head. But when they had created this model, they then used a large drug library to screen for drugs that would be beneficial or harmful for different AD signatures. There was about 30,000 compounds in that library. And one thing that was very interesting is that suvorexant actually popped out as a beneficial drug in this unbiased screen in 2 different of the mouse models, and also Ramelteon, which is a melatonin receptor agonist. We had some somologics data looking at a bunch of different proteins. I'm going to talk more about that later, but aducin-3 or ADD3 was a gene that came out of their screen as being potentially important. So we had that as part of our somologic assay, and we looked at the relationship between p-tau181 and ADD3 and showed that in the suvorexant 20 milligram group, there was this relationship where if there was more phosphorylation at p-tau181, there was increased ADD3 associated with it. So kind of translating back to humans and validating that approach. I'm very interested in looking at plasma AD biomarkers, like I said, so we've done that as well in our catheter study using suvorexant, and found some, again, very interesting findings. The left side shows CSF A-beta 40 and 42, and plasma A-beta 40 and 42, and the changes we saw in CSF did not seem to be replicated in the plasma. But when we look on the right side at p-tau181, the decrease in CSF for p-tau181 is not reflected in plasma. We actually see the opposite. So it's actually increased in blood. And this is work that Alan Lee from Washington University, who was a neurology resident, worked with me on it. And so one other way to look at this is to take our 8 a.m. to 4 p.m. and look at the area under the curve for the percent change of p-tau181. And when we did that, we found this inverse relationship where when the CSF p-tau181 was higher, it was lower in plasma and vice versa. And we were wondering what's causing this. What is suvorexant doing here to cause this effect? Just looking at this figure, there's really 2, and thinking about the A-beta data, there's only 2 explanations that came to mind as being highly plausible. One was a clearance effect, similar to what I had shown earlier with sleep deprivation. And the other is different phosphorylation effects between the 2 biocompartments. I think if it was clearance, purely clearance, I would expect to see something similar with amyloid beta. And so we performed the 11K somologic assay on CSF and plasma at hour 0, 16, and 24. So hour 0 is the baseline, hour 16 is the maximum biomarker effect, and hour 24 is right before they get their second dose, and also controls for circadian effects. And this is the volcano plots looking at these proteins. What we did is normalized hour 16 to hour 0 and hour 24 to hour 0, so looking at the change for each person. And there was, none of these individual proteins survived correction for multiple comparisons. There's 11,000 of them. There's only 25 participants that we're looking at. I think that's very surprising. But we did see in the unadjusted p-values an increase in proteins initially in the CSF, followed at hour 24 by a lot in plasma. But using all these individual proteins, we can look at different pathways. So we can do the gene ontology pathways, the KEGG pathways, and others. And when we do that, we found pathways that were significantly changed by suvorexant, even after doing correction for multiple comparisons. And so in this slide, it's the pathways that were increased by suvorexant 20 milligrams compared to placebo at hour 16, and this is in CSF. We can look at the different pathways, the interaction of these proteins, and some of the ones that we're seeing do have some overlap with what was just published from the Eric Music lab, such as endocytosis, which is highlighted here with the arrow. And then we look at hour 24 in CSF and what's increased. We see proteolysis and endocytosis, which were also some that they found in the Eric Music paper. I'm not going to go through all this data. It's unpublished. We're still kind of putting some of this together. But some of our early thoughts about this are trying to put all these different pathway changes together into a kind of a conceptual model that integrates them as to what suvorexant could be doing, at least acutely, is one, changing tau phosphorylation, decreasing it in CSF, increasing it in the blood. And we are seeing some changes in both biofluids that could account for that, as well as some clearance mechanisms, some pathway changes that we think could be associated with lymphatic clearance, endosomes, as well as some autophagy changes. And then in the blood, a lot of the changes are associated with immune factors. And our early hypothesis is that maybe the clearance of these brain-derived proteins is triggering a bit of a peripheral immune response. But I think a lot more work is really needed to be done. And speaking of some of that future work, some of it's ongoing. There are a number of trials using dual orexin receptor antagonists that are ongoing. There's two that I'm leading at Washington University and soon the University of Minnesota. One is looking at suvorexant 20 milligrams versus placebo over 18 to 24 months. And we're tracking plasma p-tau217 to see if we can slow down the accumulation of amyloid plaques. These are in participants who are cognitively unimpaired but do have some evidence by plasma 80 biomarkers that they do have amyloid plaques present. We're also looking at lemborexant for 6 months at 2 different doses, 10 milligrams, which is the highest FDA-approved dose, and then 20 milligrams. This is also in preclinical participants who are unimpaired but have some biomarker evidence of amyloid plaques. And there we're following plasma p-tau181. There's a study of daridorexant at McGill under Sylvia Villeneuve, which is following plasma p-tau217 as well, a slightly different age range and looking about a year duration. And then here locally, Professor Sharon Naismith is leading a trial that's soon to be started looking at lemborexant in MCI participants. And I think one thing that I would really like to do and find very exciting would be if we could, take data from these trials when they're completed, and potentially pool samples, maybe do harmonized AD biomarkers, omics studies, and look to see what differences are there between groups or different populations. Maybe incorporate trials like there's one of trazodone in AD, and really figure out like what are these drugs doing and how is it affecting AD biomarkers, and then looking back into the mouse and potentially then using that data to refine future studies and really advance the field. I'd like to acknowledge the participants for these trials. We do these lumbar catheter, they're very intense. We have an amazing group of participants who've agreed to work on them over the years. I'd like to thank everyone in my lab and my collaborators and all my funders. So thank you very much for your attention. take any questions that you have.
Are there any questions? Please come up to the microphone if you have any pressing. Yeah, sorry.
So thank you very much, Craig Ritchie, University of St Andrews and Scholar of Brain Sciences. I'm very fortunate. I run a clinic for former professional rugby players. Concussive and subconcussive injury seems to almost cause endemic sleep problems in former players. And I just wondered if there was a mediating effect of sleep disturbance on CTE and whether DORAs might actually be a good intervention for the retired player to prevent CTE.
That's a great question. I don't know well, one, I'm not surprised that CTE results in sleep problems. I believe that's been, described before. The exact mechanism, I'm not sure about. I think it could depend on areas that are damaged, if it involves, or how extensive it is. And it also could be the timing, like, as they get further out and they say develop pathology in the brain, is that resulting in some network disruption around sleep-wake activity? In terms of DORAs and how, what its effect could be, I'm not aware of any studies or data that's ongoing, but I do think DORAs as a pharmacologic intervention for sleep are much more preferable than most of the existing drugs, because you're not sedating the brain, you're blocking this wake signal, and it's a much more, as much as you can have, I guess, with a drug, a more naturalistic sleep onset, and has been associated with fewer side effects. I think, there's all these mechanisms that are being shown, with the phosphorylation changes and now the microglial clearance, and this is very much an evolving story. So I think as that plays out, it may be, worthwhile thinking about different mechanisms that DORAs could be causing to prevent the changes in the brain. I think another question is what could be the effect on cognition, and there has been, I didn't talk about it in this talk because it was mainly focused on AD pathology and the proteinopathy changes, but there are animal model studies looking at how DORAs affect cognitive performance in mice that develop, say, tau. Laura Jacobson, I know, has done this work here at the Florey Institute. And certainly data in human studies has also shown that it seems to be better tolerated in terms of not having the adverse consequences for cognition, falls, et cetera, that you see with other sleep hypnotics.
Thank you very much.
Any other questions?
Just one question. Carlos Roberts, old age psychiatrist from Brisbane. Thanks very much. That was really interesting. I'm just wondering if there's any thoughts on the relative effectiveness of these medications at different stages in life, knowing what we do about the changes in sleep architecture as we get older. And obviously most of the trials are being done in older people where there's some concern about lymphatic clearance deteriorating as you get older, whether we would expect much more efficacy at that age rather than in people who are a lot younger.
I think that's a very important point about looking at sleep and aging and Alzheimer's disease, is that sleep is changing across the lifespan. There's lots of well-known effects. And in terms, you asked about clearance. I think one of the, and we were just talking about this before the talk, one of the issues around studying clearance in humans has been sort of a lack of really good tools to do that. I showed some of that biofluid, AD biomarker data, which I think we interpreted as clearance being the mechanism, but it's not something that we've clearly shown based on the data that we have, and the study wasn't designed for that. So there are some tools and devices that are coming out that I think will allow for more sort of in vivo measurement of changes in clearance throughout the night that would allow us to look at different ages, different stages of AD, and how is that changing, and how a drug like this is affecting it. I do think that the the time in life at which you look at a change in sleep or you do a sleep intervention, could be critically important. My hypothesis is that when you're younger, clearance is probably less of an effect on these proteins rather than production and release. It's the release of amyloid beta and tau has been associated with synaptic and neuronal activity. That's going to be lower during sleep, during slow wave sleep, and you lose slow wave sleep as you age. But as people get older, potentially enhancing clearance, which I hypothesize is being reduced, may be a more effective intervention strategy than increasing it when they're younger. But I think it's very much an open question, and we don't have the data about how these processes are changing across the lifespan.
Thank you.
Thank you. I'm Parminder Sastry from the University of New South Wales. I was actually quite puzzled by this finding that the A-beta 42 levels went down in the blood, because my understanding was that if you're looking at clearance through the lymphatic system of the periarterial or venous wastage clearance into the lymphatic system down into the deep cervical lymph nodes into the blood, you'd probably see an increase rather than a decrease if you're making it more efficient as a clearance system. So can you explain why that is happening?
So for the sleep deprivation experiments, we were comparing sleep-deprived individuals to normal sleep controls. And in that case, it seemed like, from what we had found, that the sleep deprivation was resulting in lower levels of plasma A-beta. And so the clearance was being impaired, was our hypothesis. Now, what mechanism, I'm not sure. I mean, we were just measuring cerebral spinal fluid and blood, and we didn't really have measures and just comparing those at the same time, what were the levels. We didn't have, other measures of lymphatic function. The imaging measures are very unsatisfying in my opinion, but there's a new device that can measure the fluid flow, be electrical impedance, as well as cardiopulmonary changes, and potentially incorporating that data into studies like this may allow us to look at how clearance is changing. But our hypothesis was that since these biomarkers were going up in spinal fluid with sleep deprivation, but they were going down in the blood, that the CSF to blood transit was being impaired. Now where that was occurring, I don't know. Could it be transport across the blood-brain barrier, or absorption somewhere else, or impediment of lymphatic flow? I mean, we don't have the data to sort that out mechanistically.
Yeah. Okay. The follow-up question is actually one of the questions we're asked as clinicians by patients is, how much sleep do we need? So do you have any ideas on that?
Yeah, this is an interesting question. So, I mean, as a sleep physician, kind of the best measure I have of how someone's sleeping is essentially like, how are you feeling? And I certainly have talked to patients who report sleeping, 6 hours a night. They're concerned because they hear they should be sleeping more, but they don't have any problem that I can discern. One person I remember, I was in the Air Force at one time, and I saw this individual who was high functioning. She'd get up and run miles, at 4:30. She was high performing at work. She wasn't taking naps or tired during the day, but she only slept about 6 hours, and she just was very concerned about it. And she had no risks for any sleep disorders. And so I just provided reassurance in that case, because what are you going to do? Give them medication so they sleep longer? That seems like a insane idea. So that's usually the way that I handle this. When you look at population studies, you get these like 7 or 8 hour numbers, but a lot of those are self-report. So we have a large cohort at WashU that has self-reported sleep measures, actigraphic, which is from a wristwatch measuring movement and light, and then a forehead device called the Sleep Profiler that measures a single channel EEG, so looking at actual brain activity to stage their sleep. And we had compared the nights where we had all three data, and there's about an hour difference from the self-report to the EEG. So what they were reporting about 7 hours on average with the sleep logs, but about 6 hours on the EEG, with the actigraphy kind of right in the middle. And so you know, what people are reporting versus what they're actually experiencing, I think it becomes a question of, in terms of the length, like how are you going to measure that? And you know, how do the studies we're looking at measure it? What's very clear is as you start to get further out, you know, either people sleeping too long or too little, it is associated with worse health outcomes, either cognitive decline, mortality, heart disease, pretty much everything that I'm aware of that has been looked at, there's this U-shaped or inverse U-shaped relationship. And so what I tell people when they ask about this, because it does come up a lot, is, you know, depends how you're feeling. If you're not feeling like you're getting good restorative sleep, to, you know, potentially be checked out for sleep disorders. Because if you have sleep apnea, restless leg syndrome, or insomnia, there's a lot of treatments that are out there and available to you.
Is there time for another question?
Yeah.
My name is Johannes McKayley, and I work at the University of Sydney with Professor Sharon Naismith. My question, I guess, is in relation to the impact of these DORAs on plasma and CSF biomarker levels. You know, in this era of biomarker testing, how do we manage someone that might be on a DORA, and if they were indicated to go off for any biomarker testing, is it in the short term going to affect potentially the levels or the concentrations that we see, perhaps increase false negatives or otherwise?
So just to make sure I understand, you're asking, like, does the drug affect the plasma AD biomarkers in a way that could impact diagnosis?
Plasma or CSF biomarkers?
I mean, so the data that I presented is all hourly, so it's over just hours. You know, what could happen over weeks or months is an open question, and there's those trials that are ongoing, so we simply don't know. You know, there could potentially be, you know, an effect where, you know, tachyphylaxis, where after a week everything sort of normalizes again, and we're not sure. I think I'm most confident, although not that confident, that plasma p-tau 217 may be less likely to change, simply because it seems to be a very good marker for amyloid plaques, and I wouldn't expect that that's going to change their plaque burden over, you know, a shorter period of time.
Yeah.
But these studies are all ongoing, looking at what these drugs will do to that specific marker over 6 to 12 months.
Yeah.
And I think when they read out, we'll have a better sense. I do think that there's a lot of caution I would have just about using plasma markers. There's a lot we don't understand about them, and I read a lot of grant applications where they basically just want to convert over to looking at plasma AD biomarkers and expecting the same directionality, and I don't think that's a given, depending on what you're looking at. And, you know, potentially an increase in some of these proteins in the blood may be a positive sign, as I've kind of suggested, where you're clearing them out of the brain and lowering the CNS levels. But it's an important and really unanswered question.
Thank you.
Thanks, Joe. I'm just going to take a prerogative and ask a couple of biomarker questions. You used phospho-tau 217. Was that a brain-specific 217? Because I'm just interested in whether that you may get much cleaner data if there was brain-specific as opposed to just of the peripheral nature of some of these tau biomarkers. Second question is, have you considered other AD-related biomarkers that are not tau, like GFAP, for example?
Mm-hmm. Yeah, so to answer the first question, you know, we did the plasma AD biomarkers through C2N, and so you know, they have a validated assay that it's reflecting changes in the brain. I think the other AD biomarkers is a really important question, and with my sleep deprivation study, I first published amyloid beta, and then we had some more data, so we did tau and phospho-tau, and we published a paper, and we sort of have done sequentially all these biomarkers. So I've intentionally built into our trials that are ongoing to look at a whole panel. So, you know, I showed the primary outcomes, but we're going to be looking at multiple forms of phosphorylated tau, A-beta isoforms. We don't have GFAP as a biomarker, but we are going to be looking at soluble TREM2, we'll be looking at neurofilament light chain, and neuronal pentraxin 2, which is a synaptic marker, is planned.
Final question, Colin Masters.
A comment and a question. We're getting surprising data now looking at longitudinal change of plasma and CSF biomarkers over periods of years. And what we found, we published a few weeks ago in Annals, was the plasma A-beta falls dramatically at approximately 14 centiloids. But even more surprising, which hasn't been published yet, but we'll submit the manuscript soon, is that the CSF drops 7 years before we see the change in plasma. So the equilibrium between CSF and plasma is a very strange type of equilibrium that we still don't really understand.
No, I completely agree that there's just a lot that we don't know about how the plasma AD biomarkers change over time in different medical problems, different medications. There's a whole host of factors that could affect that, and I think some caution in looking at the effects of say an intervention in the absence of having paired samples is certainly warranted. Absolutely.
Well, I think we all agree it's a most exciting, stimulating area of research. Brendan, thank you for educating us. Please join me in thanking Brendan and Lucey. Just some housekeeping comments. Some people are having trouble accessing the abstracts, and particularly Brendan's talk, for example. So when you go to the app, go to the session, click on session for the bio, and then click on handout for the abstract. Thanks, Jerry. Thank you very much. Morning tea is waiting for you.
Hello, everyone, and thank you for joining our symposium this morning. My name is Susanne Rohr. I'm Associate Professor of Epidemiology and Social Determinants of Dementia at the Center for Healthy Brain Aging, or short CHIBA, here at UNSW in beautiful Sydney. I'm delighted to chair this session alongside my colleague Simone Reppermund, who you'll meet in a minute, a fellow associate professor at CHIBA, where we both lead research on the social determinants of aging and dementia, among other things. But before we begin, I'd like to acknowledge the Gadigal people of the Eora Nation, the traditional custodians of the land on which we meet today, and pay our respects to elders past and present, and extend that respect to all Aboriginal and Torres Strait Islanders peoples here today. And after the powerful welcome to country delivered by Brendan Karen yesterday, I would also like to follow his invitation and encouragement to look a little bit beyond these words themselves and reflect on the realities, or some of the realities they actually point to. And this means acknowledging the history and ongoing injustices that contribute to profound health inequities here in Australia. And that's especially important in the context of the symposium which we are hosting here. Mr. Karen spoke about forced family separation, incarceration, violence, and the denial and loss of cultural identity, highlighting consequences of decisions made over the heads of the people most affected by them. And as much as these are political issues, they are health issues, and they are brain health issues particularly. Aboriginal and Torres Strait Islanders peoples continue to experience some of the poorest health outcomes and highest mortality rates in their own country. And these inequities are rooted in the conditions in which people are born, grow, live, play, work, and age. Political, social, economic, environmental, cultural, and historical conditions. We are collectively referring to these in public health research as the social determinants of health. And when we talk today in this symposium about social determinants of brain health, and therefore the bigger picture of how we could and should look at dementia risk reduction and prevention, we are talking about the factors that sit beyond the individual. That is, population-level circumstances that enable or constrain opportunities for people to build and maintain a healthy brain across the life course, and thus who is more likely to develop dementia later in life and who isn't. And with that, I'd like to hand over to Simone and introduce our first speaker.
All right, very exciting. It's my great pleasure to introduce our first presenters for this symposium. It's a co-presentation by Professor Dina Logiudice and Dr. Joe Luke. And the title of the talk is Factors Contributing to a Good Spirit, Good Life for Older Aboriginal and Torres Strait Islander People.
Good morning. I'd like to begin by acknowledging Gadigal ancestors of this country and recognise the history of this place. And I'd like to also pay respects to Aboriginal and Torres Strait Islander people present. My name is Joanne. I'm an Alawa woman with my family from Central Australia, and I live in and I present Dina.
Thanks. Thank you. So the rates of dementia in First Nations populations are high, as we're all aware, up to 3 to 5 times the general population. That's a consistent finding worldwide. Potentially modifiable risk factors in First Nations populations are also higher, as you can see here in this graph that we all are aware of. And this one was modified by Rianne Lucey in Queensland, and shows that the percentage of modifiable risk factors in Aboriginal and Torres Strait Islander people was 45% compared to 38% in the general Australian population using ABS data, and compared to 32% in Asian populations. So this mirrors findings in First Nation populations worldwide, including in Canada and New Zealand. And this indicates that the risk factors are likely influenced by similar Cultural and historical factors of colonialism, trauma, things that was mentioned before, racism and social inequities. So factors such as childhood trauma and racism aren't often included in these equations. And similarly, culturally relevant protective factors are also not included. So Jo will discuss the Good Spirit, Good Life, which is a tool co-designed by elders as a framework that represents a good life or optimised quality of life that could be potentially used to explore the currently unmeasured factors contributing to brain health. So, see if that goes. The next of the star, is it? Oh, there we go. Got it. So just a couple of slides on what we do know around risk factors and social determinants in this population. So this slide shows the Let's Chat Dementia program, which is a national RCT that focused in 12 Aboriginal primary healthcare services nationwide, where a best practice dementia model of care was implemented. As part of this study, very extensive primary care audits were undertaken, over 1,600, documenting 24 different dementia risk factors, only some which are highlighted today. So the well-known ones such as hypertension, polypharmacy, diabetes, smoking were all documented as quite high levels, and we know are well documented in primary care, between 40 to 56%. Yet features such as physical activity or other social determinants such as history of childhood trauma, social isolation, etc., were very poorly documented. We also documented that about a quarter of people actually experience more than 7 of these dementia risk factors as part of the cumulative rate. The next slide, it doesn't like me, this. Oh, there we go. So there's, as we know, there's no national-based data, and the majority of knowledge comes from 3 cohort studies, and Dr. Nguyen, who's here, Hong, as part of her PH study, actually combined these 3 studies from the Kimberley, from urban regional New South Wales and from northern Queensland, and harmonised the data for 900 First Nations people over the age of 45 or 60. Most information is cross-sectional, but 40% was longitudinal. So we can see here that protective factors was education. Risk factors longitudinally were age and hearing impairment, and some of the cross-sectional factors, again, as we would expect, stroke, high rates of head injury, epilepsy and antidepressants were also significant risk factors. Unfortunately, data on factors such as physical activity, social determinants were limited and inconsistently available in these cohorts. So limiting the ability to expand on work from New South Wales Koori Growing Old Well study that identified that unskilled work was a significant risk factor in longitudinal data, and a history of childhood trauma and low physical activity also in cross-sectional. So this is an area that requires new understanding as we learn about more meaningful cultural and social determinants of health that Jo will discuss. So I hand over to Jo. Thank you.
So you're probably aware of the World Health Organization definition of the social determinants that describes the non-medical factors that influence health outcomes. They are the conditions in which people grow, are born, work, live and age, and the wider set of forces and systems shaping the conditions of daily life. These forces and systems include economic policies and systems, development agendas, social norms, social policies and political systems. In considering this definition, Aboriginal and Torres Strait Islander people's social worlds are more than just socio-economic circumstances. Aboriginal people are born, grow, work, live and age in Australia where structural determinants such as colonisation and racism shape health. They are also born, grow, work, live and age into communities where cultural determinants such as connection to land, country, family, community and languages are also important to living a good life. As part of our recent research, we looked at cultural determinants for older Aboriginal and Torres Strait Islander people and show how the social lives are not always the same as other Australians. This graph shows some of the unique activities and events that Aboriginal and Torres Strait Islander people participate in. Some of these include being involved in organisations, NAIDOC week activities, ceremonies, fishing, hunting, arts, music, and also storytelling and passing on stories. If we consider the data on the social determinants and dementia that Dina spoke to, these studies were largely from a Western frame that measured dominant social determinants, for example, childhood trauma and education. We believe more accurate and understandings will come when we take a more inclusive understanding about the social determinants of health, which also attend to the structural and cultural determinants. We think a good starting point for future research would be the Good Spirit, Good Life framework. This is a quality of life framework that was developed with input by older Aboriginal and Torres Strait Islander people, and it really speaks to their social worlds. This framework was developed by Smith et al. Professor Dawn Bessarab had a large input into this one. And it talks to the 12 factors that Aboriginal and Torres Strait Islander older people identified as important for them ageing well and having a good spirit, good life. Some of these, for example, included safety and security, the important role of elders, connection to country, community and culture. By looking at the diverse social worlds of Aboriginal and Torres Strait Islander people, we will then be able to have a more nuanced understanding about the risk and protective factors for dementia for this population that are upstream. So in concluding, the social determinants of health, inclusive of cultural, commercial, structural, contributing to dementia in Aboriginal and Torres Strait Islander people are poorly identified and documented. And we believe that further research really needs to take a more inclusive view of the social determinants. Yes.
Thank you very much for this very powerful talk. Are there any questions? I might start. So you talked, there is not a lot of data on in the Aboriginal Torres Strait big community feeling, and so I wonder, how would a study look like? Sorry. I was just saying that in individual risk factors, we see that social isolation and loneliness is a big risk factor. And I wonder, with a rich Aboriginal community gathering, what is the data, or is there any data on this?
I guess in terms of epidemiological data, there's largely national data sets like the National Aboriginal and Torres Strait Islander Health Survey and Social Survey. They include some measures around connection and involvement. That's where that data that I presented came from. But more specific ones related to ageing.
So I think it's an area that needs to be defined, what should be measured, because there's commercial determinants, there's social determinants, there's community determinants, environmental determinants, etc. But it really needs to be come from the voice of Aboriginal people.
You want me to go first? Great talk, thanks. I'm really encouraged by the emphasis on a strengths-based approach and wondered if perhaps when you talked about the risk factors, maybe if you could elaborate on some of the strengths also that Aboriginal communities will have as well.
Yeah, and that's a great question, Bianca. So one of our colleagues, Jessie Zanka, is a geriatrician, and his focus is on frailty. And Aboriginal communities have been saying, this deficit approach is not appropriate for communities, and Joe will be better at explaining that. And it will be important, whatever we look at needs to come from that strength-based approach. So he's actually looking at a frailty using a strength-based lens. But yes, thank you, that is very important. And that's where the Good Spirit, Good Life is so important, because it does show the areas that do optimise well-being for an older person.
Run out of time for questions. Thank you very much for your talk. All right. And the next speaker is Dr. Annabel Matison from the Centre for Healthy Brain Aging, UNSW, and her talk is on neighbourhood environments and transition to cognitive states from the Sydney Memory and Aging Study. Welcome.
Thanks, Simone. So good morning, and thank you for the opportunity to present. I'm Annabel Matison, and I'm actually presenting on behalf of Esther Susanne. I'm presenting our work on findings from the Sydney Memory and Aging Study on neighbourhood environments and transitions to cognitive states in older adults. So I think we're probably all aware that it's estimated that up to 45% of dementia cases are potentially modifiable. However, most of the research on dementia prevention has focused on individual level risk factors such as lifestyle and health conditions. Neighbourhood environments, which shape many of the individual behaviours and exposures, have been comparatively understudied, particularly in longitudinal research. Importantly, environmental exposures are not randomly distributed. They represent broader social and structural factors, making them an important social determinant of health. So understanding how neighbourhood environments relate to cognitive ageing may help identify population opportunities for prevention. Our study used data from the Sydney Memory and Aging Study, which was a population-based cohort of over 1,000 adults aged 70 to 90 years. Participants were followed for up to 13.7 years, and cognitive assessments were conducted approximately every 2 years. So neighbourhood exposures were measured as a 1 kilometre buffer around the participant's home, and it captured built environment, natural environment, and pollution. These exposures were assessed repeatedly across the 13.7 years, and participants were tracked each of the 2 years, allowing us to examine both short-term and cumulative environmental exposures. So for our study, we used a multi-state model to estimate transitions between different cognitive states. So, for example, transitioning from normal cognition to dementia, or transitioning from normal cognition to MCI, etc. Okay, explaining the table before we get into the details. So for this presentation, in the interest of time, I'm going to focus on the cumulative exposure, but the results for the short-term exposure were broadly similar. In the table, you can see that there's an overall model which adjusts for socio-demographic factors, which included the area level, SES, and other environmental confounders, whereas independent models additionally adjust for other environmental features which may act as mediators. So looking at the built environment, we can see a general pattern where population-dense, connected and service-rich neighbourhoods have better cognitive outcomes. So for example, population density, greater street connectivity and better access to commercial services and public transport were often associated with lower likelihood of transitioning to worse cognitive states. One important thing to note here as well is that the associations were often stronger after adjusting for other environmental features, such as air pollution. So it reflects the importance of looking at all these factors together, rather than looking at one individual factor in isolation. So overall, these findings suggest that more walkable and connected neighbourhoods support activities like physical activity, social engagement, and are beneficial to cognitive health. The natural environment. So tree canopy had the most consistent relationship with better cognitive outcomes. Higher levels of tree canopy were associated with lower transitions to dementia and from normal cognition to MCI. There are several plausible mechanisms for this: reduced air pollution, shade, and greater opportunity for outdoor activity. So in contrast, parkland showed more mixed associations. So that potentially reflects that parkland is a more heterogeneous measure, encompassing a wide range of environments which have different qualities and usability for older adults. Air pollution. So findings from air pollution were less consistent overall. For air pollution, higher cumulative exposure to PM 2.5 was associated with a higher likelihood of transitioning to dementia, particularly in cumulative models. However, associations for NO2 were mixed. So one important contextual factor is that pollution levels in this cohort were relatively low, and there was limited variability amongst the cohort. So it's possible that the exposure levels to pollution here were below thresholds that we would expect to see stronger effects, and so these findings should be viewed with caution. In terms of pathways, as I've mentioned, there are several plausible pathways linking neighbourhood environments to cognitive health. They include physical activity, social engagement, and cognitive stimulation, as well as reduction of stress and exposure to less environmental hazards. There are a number of limitations that need to be considered when reviewing these results. So first, this is an observational study and we can't infer causation. Secondly, and importantly, you can see from the graph, this was from a limited geographical area in Sydney's eastern suburbs. They were a high socio-economic population, and this may limit generalisability. Clearly, there's attrition over 13.7 years in a cohort of this age. We've tried to account for this through inverse probability weighting, but yes, it is a limitation to the study. As I mentioned previously, there was limited variability in air pollution, and the other important factors are we obviously have data on these people during the study, but we haven't got data for where they lived before the age of 70 or before they entered the study. And as well, we don't know how much of the time people actually spent in their neighbourhoods. These could be people there are obviously people who spend more time close to their home, and there are other older adults who spend a lot more time away from their homes. In terms of implications, the findings suggest neighbourhood environments play an important role in cognitive ageing, particularly features such as walkability, access to services, and urban greening may be relevant to contribute to cognitive health in older adults. These are modifiable structural behaviours that operate at the population level, and it highlights the potential for urban planning and design in dementia prevention. It also enforces the importance of addressing environmental inequities as part of broader efforts to promote healthy brain ageing. So finally, I'd like to acknowledge the participants from the Sydney Memory and Ageing Study and my co-authors and collaborators on the project.
Thank you, Annabel. Great talk. Are there any questions? Yeah? You can ask both.
Thanks very much. Thanks for such an important bit of work, and it's really wonderful to hear about some attention being paid to the environmental factors particularly. My name's Pauline, I'm from Tasmania, so I'm very interested in natural spaces. What I wanted to ask you about was where you see this approach being applied for people in a post-diagnostic space. I'm really interested in moving away from the sort of barrier between prevention work and then post-diagnostic. It feels like you're doing a lovely little bit of work here about transitioning from normal, functioning brain health or whatever to MCI dementia. I wonder about the next stage, living well with dementia, and the importance of these factors and how you see this work being applied in that area.
Yeah, it's a good question. We actually were asked by a group of, I can't actually remember the name of the magazine, but it was an aged care to provide them with input in terms of what could be done for, well, their residents in general. So it's kind of around setting up aged care to, have more green space, be closer to cafes where families are likely to take the residents out. Not be next door to the main road. All of those kind of things should be fed into where a person lives. And it's not always easy to move house, move to an area which is greener. But trying to factor those kind of things into the thinking process.
Hi, my name's Hannah. I'm also from Tasmania. I was wondering about your measures of, say, tree canopy cover and blue spaces and how you either measured or where you found the data on how much canopy cover was within a kilometre of a person's house.
Okay, so I can't tell you exactly how that was all done because that was done by a group of people that Esther was looking after. But essentially, in terms of we had their residential address, and we did it 1 kilometre walking distance, so using the street network, and I believe it was like GPS data that looked at the tree cover and blue space. But don't exactly quote me on that one.
Right, thank you very much, Annabel. And I'd like to now hand over back to Susanne, who will also be our next presenter.
Thanks, Simona. Briefly switching my head from chair to presenting myself. And I will share some findings from a secondary analysis of the Maintain Your Brain trial, asking an important follow-up question, and that is, when an intervention works, does everyone benefit equally, or does it actually lead to widening or narrowing social inequalities in cognitive outcomes? The reason why it's important to ask this question is that it is quite well established that dementia risk and cognitive functioning are not equally distributed across populations. We've heard a bit about that earlier today. And likewise, we know that a substantial proportion of dementia risk is attributable to potentially modifiable risk factors. But these risk factors are also not randomly distributed, but follow a social gradient. So, for example, people experiencing greater socioeconomic disadvantage are more likely to be exposed to dementia risk factors across the life course and experience poorer cognitive trajectories and ultimately face a higher risk for dementia. The figure here on the right illustrates this using data from the German national cohort. Here, across adulthood from age 20 to 75 years, we see that individuals with lower socioeconomic status consistently accumulated a greater burden of modifiable dementia risk factors than those with higher socioeconomic status, and all of that linked to cognitive outcomes. And this is why when we deliver interventions addressing modifiable dementia risk factors, we need to ask, do they help reduce inequalities, or do they actually reinforce them? So multi-domain lifestyle interventions are now increasingly being recognized as a promising approach to dementia risk reduction. The approach was pioneered by the Finnish finger trial, which demonstrated for the first time that a multi-domain intervention could improve cognitive function in older adults at increased dementia risk, and kind of like validating the five-finger model, suggesting that targeting nutrition, physical activity, cognitive training, social activities, but also vascular risk management is quite good if addressed simultaneously to benefit cognition. Evidence remains a bit mixed if you look at all the trials that are coming out, but more and more are showing indeed small to moderate benefits for cognitive functioning. And more recently, the digital delivery of such interventions has attracted quite considerable interest because it offers the possibility of reaching larger populations at scale and relatively low cost compared to face-to-face programs. However, there may be a downside. There's quite a bit of literature that suggests that digital interventions may actually widen inequalities and have inequity effects. So if aspects such as participation, engagement, adherence, or benefit differ systematically across social groups, then that's obviously nothing we want to achieve. And currently, we don't have any such evidence for multi-domain interventions for cognitive outcomes. To address this gap, we drew on data from the Maintain Your Brain trial, or MYB. I assume MYB is quite well known among many here in the audience. Just for a little bit of context, it was a large Australian, fully online delivered multi-domain randomized controlled trial involving more than 8,000 adults aged 55 to 77 years at elevated risk for dementia. And it targeted 4 major modifiable risk factors through digitally delivered modules focused on physical activity, nutrition, cognitive training, as well as mental health. Participants were assigned to the intervention group, received personalized coaching, tailored content, reminders, behavioral support, depending on the dementia risk they brought at baseline, and participants in the control group just received static information based on the different lifestyle factors. And as shown in the primary outcome publication from last year, the intervention did indeed result in a significant improvement in global cognitive functioning over 3 years. So MYB worked overall, and now we are asking whether those benefits were equitable. We investigated whether the intervention modified cognitive outcomes across 3 indicators of social inequality: area-level socioeconomic disadvantage, years of formal education, and individual-level socioeconomic status, combining education, household income, and occupational status. The outcome was the same as in the primary analysis, a global cognitive composite score, and we drew on an inequality difference in differences, or DID framework. So we compared specifically cognitive trajectories between participants at the 10th and 9th percentile of each social indicator, so basically the margins of the population, and examined whether this gap became larger or smaller in the intervention group compared to the control group. And a negative DID estimate would indicate a pro-equity effect. So what did we find? After 3 years, we actually observed quite an encouraging pattern. There was no evidence, and this is really important here, that the intervention benefited more advantaged participants more, which is obviously an often expressed concern for digital health interventions. Instead, we actually found a significant reduction with regards to socioeconomic status. The cognitive gap was reduced by approximately 0.22 standard deviations in the intervention group compared with the control group, and that is a meaningful finding, especially if you think about rolling this out at the population level. And we also observed a small significant reduction in inequalities related to area-level disadvantage, but not so for educational inequalities. But if you look at the panels, you do see all estimates pointed in the same positive direction when it comes to pro-equity effect over time. Okay, if we look at these figures, we first know that cognitive performance improved across all groups, regardless of social position. But the second observation is that the intervention appears to have generated relatively larger gains among participants experiencing greater socioeconomic disadvantage. And again, most evident for individual level socioeconomic status shown in the right panel. Now, to better understand these findings, we conducted a series of all sorts of different additional analyses. So first, it's really worthwhile to note that participants from more disadvantaged backgrounds entered the trial with a higher burden of modifiable dementia risk factors, and as a consequence, they were eligible for a greater number of intervention modules. Second, we tested the robustness of the findings using several sensitivity analysis, but the overall pattern remained quite consistent across alternative analytic samples, inverse probability weighting to account for some missingness, and all sorts of other checks. Also worthwhile to note, attrition over 3 years was substantial. That's again normal for large-scale digital interventions. However, when we run a completer analysis, it yielded quite similar effect estimates compared to the intention to treat analysis, so that these findings were not driven by dropout. And finally, we looked at engagement with the intervention. Again, there was no meaningful social gradient in engagement. It means there was no difference between individuals with higher or lower social disadvantage when it comes to how much they engaged with the intervention content. And so this also suggests that a differential uptake of the intervention is not likely to explain the observed findings. Now, to conclude, overall, I think these findings are really encouraging, both from an effectiveness but also equity perspective. Again, no evidence that the intervention widened cognitive inequalities, which is the core message here. And if anything, participants experiencing greater social disadvantage appear to benefit at least as much, and in some cases even more, than their more advantaged counterparts. One possible explanation is the risk-based tailoring approach used in MYB. Participants with a higher burden of dementia risk factors qualified for more intervention modules and therefore received more intensive intervention exposure. And importantly, all the higher risk participants were disproportionately drawn from the more disadvantaged groups. And lastly, at the same time, it's important to say what the results do not tell us. So our analysis examined inequalities across the social gradient within reach, meaning within the MYB population. The most socially disadvantaged individuals were less likely to participate in a trial in the first place. They were drawn from the 45 and up study, and therefore anyone within the lowest groups, or lowest groups in terms of epidemiological terms, were not represented in MYB. And it's also important to note that MYB was delivered free of charge within a research setting. So now when it comes to thinking about implementation, there was just the funding from the MRFF to roll MYB out, which is fantastic, but then you need to look at financial barriers, technological requirements, digital access, health literacy, etc., which all could influence equity under a real-world scenario. But financial accessibility was probably one of the drivers here. So the challenge is now to ensure that interventions like MYB remain accessible, affordable, and capable of reaching those who are currently the least likely to participate or benefit from prevention efforts. But I guess for now, we see some really positive signals. So with that, I would like to thank you all. I'm not sure if you have time for questions, but Simone will let me know.
We have 2 minutes. So we would have time for 1 or 2 quick questions.
Thank you. I have a question. That's really promising data that you're sharing. Can you talk a little bit about the design of the intervention and how you tried to build in accessibility so that everyone could access the information equally?
Right. So during the trial, individuals were assigned in the intervention group, 2 modules depending on the dementia risk they presented at baseline. So for example, a person would only be assigned the module to do physical activity if they were physically inactive, or if they were only doing the nutrition module, if they were showing patterns of not ideal nutrition. So it was a stratified approach, which I think is really important here. So accessibility was sort of gate-kept according to what a person needs. And this needs-based approach is a differentiator here. So during the trial, obviously everyone had access. There was no barrier with regards to paying money for accessing, the online modules. It was presented on either laptop, computer, or tablet. It was not a smartphone-based delivery. And yeah, I hope that answers the question. Sure.
Hi, thank you so much. That is a lovely research, especially because a lot of the times I see all of these studies that are inaccessible because people have to travel. I was just wondering, in terms of confounders, what did you control for? And one of the things that I believe it's important, but I haven't seen that it's mentioned, is did individuals that were participating in doing these modules have someone to help them? Because there are a lot of cases that older individuals receive help for tech from their children or a carer or someone that lives with them.
Yeah, great question. So the intervention was mostly self-guided, so there was no professional or health support, which was an intentional design to make it scalable and more cost-effective. We cannot say if spouses, carers, or whatsoever supported a person when they were going through a module, but I wouldn't say that's a problem in a real-world setting, as well. If someone supports a person to adhere to whatever has been suggested to do in terms of exercise. And what was the other question?
I was just wondering what confounders.
Very good question as well. So we drew on the analytical framework of the primary analysis, because obviously you want to interpret the findings in the context of that. And they did unadjusted as well as adjusted analysis, and what they call fully adjusted model included age, sex, education. Education, obviously, we didn't do for looking at education. Number of eligible modules and modifiable dementia risk, which was operationalized with the ANU ADRI dementia score.
Thank you so much.
Thank you. All right, it's now my pleasure to introduce our final speaker, Professor Blossom Stephan. Professor Stephan is the inaugural chair in dementia for Dementia Australia and Curtin University in Perth, as well as the director of the Dementia Center of Excellence at Curtin. And she will discuss how social determinants can be better incorporated into dementia risk modeling and highlight opportunities to strengthen evidence from low and middle income countries, moving away from Australia to a broader international context. Over to you.
Super, thank you, Susanne, and thank you so much for the opportunity to present today. So in this presentation, I'm really going to highlight how dementia risk prediction and prevention research really isn't targeting or reaching those communities that stand to benefit most, and how we can address this gap. Oh, there we go. So starting and setting the context. So we know dementia is unequally distributed across the world, with roughly 60 to 67% of dementia cases residing in lower middle income countries, and this number forecast to reach 71% by 2050. And this statistic and this number is really meaningful because it's these communities that have the greatest burden of dementia, but had this challenge within the context of really limited infrastructure, limited healthcare budgets, and limited services for prevention, risk reduction, and even diagnosis and the follow-on care for people living with dementia. So if we're going to be able to prevent dementia, we need to know where we can make the greatest impact. So looking at 7 of the 14 Lancet Commission risk factors, so low education, physical inactivity, hypertension, obesity, smoking, depression, and I think it's diabetes, I can't really read it. We can see that the combined population attributable risk for these 7 factors really varies across the world. So if we look at the West Pacific data, we can see that the range of attributable risk for these 7 factors ranges from 20%, just over 20% in Singapore, through to the highest in the West Pacific region at 34.7% in Papua New Guinea. But the highest combined attributable risk we see for this is actually in the Portuguese data. So this suggesting that income country tier really isn't predicting where the greatest burden is or where we need to focus our intervention strategies. And if you look at somewhere like Australia, a high income country, has a combined attributable risk of 25% versus Cambodia in West Pacific, which has 22%, so very small difference across these 7 risk factors. So that said, if we combine across income levels, so high income versus lower middle income countries, we actually see that there are some risk factors that have a greater attributable risk. So in lower middle income countries, we see nearly doubling around low education, physical activity and socialisation in lower middle income countries, whereas in high income countries, we see high levels of hypertension and diabetes really driving risk, and this is probably due to the prevalence of these conditions and our ability to identify them and treat them. That said, where the research so far has really focused on health and lifestyle risk factors and is really missing the social determinants of health. So last year, Seb Walsh published a systematic review identifying 6 domains covering the social determinants, 3 which has strong evidence around educational attainment, physical environment and pollution, and income, work and socioeconomic status. 1 that has moderate evidence around social inclusion and ethnicity, and 2 that are emerging, particularly around food environment, so access to different foods and dietary patterns, and then housing and sanitation, so limited evidence on homelessness and incarceration. And what we're not seeing within the dementia risk prediction or risk prevention research is incorporation of these risk factors. So what are we doing within the risk prediction domain? So here on the slide on the timeline, I've included 5 prediction models that have been developed using an evidence synthesis approach, so the Australian National University Alzheimer's Disease Risk Index, or have used data from high-income countries. So you've got the CAGE model from Finland, HCOD from Germany, the BDSI from the USA, and the BDRM from the Rotterdam study out of the Netherlands. And I've chosen these models because they have been validated in both high as well as lower middle income country settings. And we can see that they really overlap in terms of their predictive variables. So they all include age and sex, and then they include measures of lifestyle factors and health factors, namely around cardiometabolic disease, and some also include cognitive and physical function. So what happens when we take these risk models and we map them in lower middle income country settings? So these are results from the 1066 study, covering 7 lower middle income countries in Latin America, Caribbean and China. And the graph shows the distribution of the predictive accuracy of each model across the 7 lower middle income countries, with the star indicating how the models performed in their development sample or in the high income country. And we can really see poor performance when we map the CADE or the HCOD, so the Finnish or the German model. And we see better performance when we map the ANU model, the BDSI and the BDRM model. But we're still seeing that the results aren't transporting clearly. You get wide variability in predictive accuracy of those models, suggesting that we need something more in lower middle income country settings. So then the advice would be to, how about we develop models in lower middle income country data? And our PhD students synthesised the risk prediction literature and identified only 5 models from 2 countries, Mexico and China, that have been developed using lower middle income country specific data. They typically incorporate variables that have been incorporated into high income models, so variables such as social demographics, education, vascular and lifestyle risk factors, and they have pretty good predictive accuracy, so we see a range of 0.7 to 0.94. But what we aren't seeing is we're seeing that they're missing those core social determinants, socioeconomic variables, low birth weight, healthcare access. So our PhD student, Sun Yangon Gayu, has taken the 1066 data set and attempted to develop a simple prediction model incorporating age, social engagement, physical inactivity, hypertension, and difficulty handling money. And across those 7 lower middle income countries, had a predictive accuracy of 0.75. And he's then added in food insecurity, late life residence, and household assets, and actually found that those variables don't enhance predictive accuracy to that simple algorithm. And it could be that at this stage of life, in later life, that these variables aren't as important as the health and lifestyle variables. So that's the predictive literature. How about the preventative literature? So affecting the population and trying to shift risk at scale. So we've just got a review that's been accepted in Lancet Healthy Longevity that looked at 12 studies with 4 delivery modes, including mass media, digital education, community programs, and critical arts-based programs, and whether or not they enhanced dementia knowledge and motivation to minimise dementia risk. And we found mass media had broad reach but limited gains or effectiveness. We found really that community-driven and educational-driven strategies were very good at changing behaviour and increasing knowledge. But we do see that lower middle income country gap with only 2 studies conducted in middle income countries, including Chile and China. So in conclusion, in terms of what we see in high income compared to lower middle income country, we see that the burden is uneven. We see that the research is really high in income country centric, and we know that in order for us to reduce the inequity in brain health across the globe, we really need to develop context-specific, culturally intelligent strategies so everybody has the opportunity to have healthy brain ageing. And thank you to all my collaborators.
Brilliant talk. Thanks, Blossom. Any questions for Blossom?
Thanks, Bloss. I think that the work that was done on the intervention, so the education, the mass media, et cetera, are also important to make sure that when we're deploying the interventions, they're appropriate to the country. So I just wanted to revisit, just in case it slipped through and you know where I'm going, on the mass media piece. And so that piece of work that said mass media has limited reach also reflects the fact that there's actually been not a really sustained investment into mass media campaigns anywhere globally, and that they've been underfunded. And so in response, you know, that's a dose response effect because they're underfunded. So I think looking at interventions, we need to look at what that intervention was in that country and was it at scale. That's my only comment.
No, Tanya, 100%. So what you see is that these are typically run on very small budgets when the budgets are reported. And where you have multi-centre sites, we tend to see that amazing reach. So in the Netherlands, they had 7 sites in the Netherlands, and you could see that it reached over 30,000 individuals who had seen the campaign. And if we don't have sufficient budgets, then we're not going to be able to reach the populations that we really need to reach most. But you're right, it will come down to not only what the strategy is, but how well it's funded. And we need the federal government to adequately fund these campaigns like they do for heart health.
We have one more question over there, please.
Hi, thank you, Blossom, for a wonderful presentation. I was just wondering, with the 5 factors model, was that age stratified? And when you added the other factors, was there any difference between kind of a midlife inclusive cohort versus an older population?
No, unfortunately, so it was based on the 1066 sample where every individual is age 65 years and over, and age was incorporated into the model. And we think that with these results, that if life expectancy is a lot lower in lower middle income countries, so the individuals who are surviving into this study are going to be naturally the more healthy, more conscious of their health, and that survivor effect And I think that's why, if they do have those adverse, risk factors, they do have the poverty and the food insecurity, they've managed to survive into later life, and that doesn't become so important as their health and lifestyle status.
Yeah, thank you.
Thank you again, Plus. And with that, we come to the end of this symposium. I would like to sincerely thank all our speakers for their excellent presentations and insights. I guess if there's one takeaway from this session, it's that brain health and dementia risk is not only shaped by choices people make, but by the conditions societies create for making choices. I would also like to thank my co-chair, Simone, and conference convener Stephanie Ward and the scientific committee for providing a platform for this discussion. And of course, thank you to all in the audience for your engagement and interest in the topic. It's time now for coffee and tea, and enjoy the rest of your conference. Thank you.